In this video, we'll be learning about gram staining. So what is the aim of gram staining means? To differentiate the gram-positive and gram-negative bacteria based on their cell wall. Okay, we all know that cell wall is made up of peptidoglycan layer, and gram-positive bacteria has a thick cell wall and gram-negative bacteria has a thin cell wall. Okay, so based on the cell wall composition, we can differentiate gram-positive and gram-negative bacteria. Okay, so in this gram staining process, we'll be using certain dyes which can stain the bacterial cell. Okay, let's see one by one and its principle. So the basic principle of this gram staining is it stains the bacterial cell wall into purple color if it is gram positive bacteria. If it is negative bacteria, it will be in pink color. Okay, it will appear in pink color. Okay, so the ability to retain the purple color or ability to retain the crystal violet, based on that we can differentiate this gram positive and gram negative bacteria. Okay, so the first dye or the first stain we'll be using here is crystal violet. Okay, so this is called as the primary stain, that is a basic dye which is positively charged. We'll take a bacterial smear and then we'll be using this crystal violet, 1 or 2 drops, and we'll be pouring on top of the bacterial smear. And what happens there is this crystal violet will get adhered to the cell membrane of both the gram positive and gram negative bacteria. It doesn't mean that it will only stick on to the gram positive. At first it will stain both the gram positive and gram negative bacteria. Okay, the cell membrane of the gram positive and gram negative bacteria. Next we'll be using the mordant. Okay, so that mordant is called Grams iodine. So this Grams iodine acts as a mordant which increases the staining ability. As we use the crystal violet here, so this Grams iodine will increase the staining ability. Okay, so the iodine fixes crystal violet into a cell wall of the bacteria by acting as a mordant. By acting as a mordant, this Grams iodine will fix the crystal violet on in the bacterial cell wall. Okay, it's just a fixation agent. Okay, remember that it acts as a fixation agent, so it will fix the crystal violet in the bacterial cell wall. We all know that gram positive bacteria has a thick peptidoglycan layer, so that it will get adhered to the iodine. Okay, so as the gram positive bacteria has a thick peptidoglycan layer, it is easy for the iodine to get fixed by the crystal violet. Okay, and stains purple. As in the case of gram negative bacteria, it consists of a thin membrane, till cell membrane, so the adherence of iodine is not present. Okay, iodine cannot adhere. Okay, soon after adding that Grams iodine, we'll be using an ethyl alcohol, which is considered to be a decolorizer. Okay, as in the case of gram staining process, we'll be using Rimmel gram decolorizer, which consists of 95% of ethyl alcohol. Okay, it's just a decolorizer. If the crystal violet and Grams iodine is not fixed, it will just decolorize. Okay, see, as in the case of gram negative bacteria, the peptidoglycan layer is very thin, so that it gets into the peptidoglycan layer and just decolorize the cell wall. Okay, decolorize the crystal violet. So that is what said here. So it penetrates deep inside the cell wall. This alcohol will penetrate deep inside the cell wall. As the cell wall of gram negative bacteria is thin, the cell wall of gram negative bacteria is thin, so it decolorizes the crystal violet and iodine complex. Okay, then due to the disability of retaining stains, it appears colorless. It will make the crystal violet colorless. After adding this decolorizer, it will make the bacteria to decolorize and appear colorless. Okay, if it is gram positive, it will not change its color. Okay, if it is gram negative bacteria, when we add ethyl alcohol, that both the grams iodine and crystal violet will get decolorized and appears colorless. Now this colorless bacterial smear, after adding the safranin is a counter stain which is positively charged and gets bind with the negatively charged compounds of cell wall. Okay, so safranin is basically a red color. So it is used to stain the gram-negative bacteria. As I said already, safranin is used to stain gram-negative bacteria. It is also a basic dye which interacts with the negatively charged compounds of the cell wall. Okay, instead of the safranin, we can also use dilute carbamazepine. Okay, this is also can be used as an alternative for safranin. Understood. Now let's see what are the materials required here. We'll have to use a glass slide and a bacterial smear. We have to prepare a bacterial smear in a glass slide, and we'll be using a primary stain, a mordant, decolorizing agent, counter stain. These are the major material required. Okay. Now let's see the difference. What are the difference appearing when we do the staining process? See, this is the gram positive bacteria, how it reacts with the stains. Okay. Now this is the gram negative bacteria reacting with the stains. Okay. Now we'll be taking both the, we'll be taking grams, bacterial smear, and we'll pour a crystal violet. Okay, now we'll be pouring crystal violet on top of it, and we have to wash it, or we have to rinse it with the distilled water. Then we'll be adding iodine. See, after iodine also, adding iodine also, this gram positive bacteria will be in purple color. Okay, for what we are adding this iodine means to fix the crystal violet with the bacterial cell membrane. Okay, now we'll be adding decolorizer. If it is gram positive bacteria, it will not decolorize. Okay, same, it will appear in the purple color. And after that, we have to rinse it with water, and we have to add safranin, which is a counter stain. Okay, after adding counter stain also, it will be in purple color only if it is positive, gram positive. See, as in the case of gram negative bacteria, we'll be taking bacterial smear and stain it with crystal violet, then rinse it with water, then add grams iodine. See, after adding grams iodine, it will just fix the crystal violet with the cell membrane. As it is mordant, it will just fix the crystal violet to the cell membrane. Okay, then we have to wash it with water, then we have to wash it with decolorizer. See, if it is gram negative bacteria, it will decolorize all the stains and appear to be colorless. Okay, then we have to add safranin. After adding safranin, this colorless bacterial smear will appear purple in color. Okay, so this is the procedure we are following here. So we know the difference between gram positive and gram negative bacteria, and the result will be spore forming bacteria with rod shaped will be in gram positive, non-spore forming will be in gram negative bacteria. Okay, so this is all about the gram staining procedure.